Well, good morning, everybody. I appreciate you taking a little bit of time to hear about our new Human Transcriptome Array. We will be taking questions at the end of the conference, just to maintain the flow. So if you do have questions, please hold them until the end of the conference, and we'll have an operator available to assist you in asking those questions at the end. So without further ado, let's get started into the presentation. So today, what we'd like to talk about is a complete application at looking at both gene expression and alternative splicing. So we'll start with a brief overview of Affymetrix, and then we'll talk about the three components that comprise the complete application, or the complete solution for understanding the transcriptome. It starts with the assay and the reagent kits. And there are two major assay options for this array. Then we'll talk specifically about the array itself, talk about some of the details of the array. And finally, we'll conclude with the analysis portion. Affymetrix has designed software to do the analysis for this particular array. And so we'll conclude with a brief discussion about the software itself. So the Affymetrix GeneChip technology was invented in the 1980s and commercialized in the late 1990s. So Affymetrix has been around for quite a long time now in the microarray field. We have products to analyze DNA, RNA, copy number changes, and these are all across the whole genome level or whole transcriptome level. There's over 28,000 publications citing GeneChip technology, so you know that it's proven, tried and true. And again, lots of platforms to address various issues, throughput, format needs, and sample types. So this is just a quick snapshot about the new Affymetrix. I'm not going to spend too much time on this, but I wanted to just introduce you to what Affymetrix is today. Affymetrix is a complete company encompassing DNA and RNA profiling and expression, and also further down the line to single-cell validation and functional analysis. And beyond that, we also generate our own reagents and assay kits to support our workflows. So it's really an integrated portfolio for a complete view of systems biology. But today we're going to focus specifically on gene expression or gene profiling. And it's important to think about how to choose the right technology. We're well aware that there are several different types of technologies out there for you to choose from, including whole genome microarrays, quantitative real-time PCR, gel-based methods, branch DNA, and next-generation sequencing. And all of these technologies are very good, and they provide excellent data. It just depends on your particular type of study, which is the appropriate platform for you. So I'd like to talk about a few of the parameters or a few of the factors that you should consider when you're trying to determine which type of platform or technology to use. So first at the top, we'll start at the top and kind of work our way clockwise around the circle here. So the first one to consider is number of genes. Are you looking at one or two genes in a small single or 2 or 3 plex type of scenario, or are you looking to go across the entire transcriptome? The second parameter to consider is number of samples, and this talks to the throughput of the technology. So if you're doing 1 or 2 samples, then there are certain technologies and applications that are applicable. If you want to ramp up and you're doing a large-scale study, 20, 30, or even more than that, then Arrays is a highly scalable platform that can work from a single sample all the way up to hundreds of samples at a time. And then the third factor to consider is types of samples. So are you using RNA derived from blood, from tissue, from FFPE blocks, formalin-fixed, paraffin-embedded blocks? So Affymetrix has assays that can encompass and work with all of these different types of samples. And then access to instrumentation is also important. Affymetrix has a very large installed instrument base. So in general, you're probably very close to an Affymetrix system. There's probably one at your institution. So access to instrumentation is very easy with the Affymetrix platform. The instrumentation is the same for all of our cartridge arrays. So if your facility or institution has a cartridge scanner, chances are you'll be able to run this assay on the existing instrumentation. And then obviously time to results is also very important. So we live in a world where we need to get to the results quickly so that we can publish or move on to the next grant and get funding to continue your study or do follow-up validation. And so getting to those results to be able to move on to the next step is very important. And being able to move to those results in a very quick and efficient manner is something to consider when you're considering which platform to choose. And then obviously amount of starting material is something that's also very, very important. If you're using cells that you can grow up in culture, then you probably have tons of RNA. But if you're using formalin-fixed, paraffin-embedded blocks, those tissues are very precious, and you don't want to use very much. And so Affymetrix has solutions that will work with a wide variety, a broad range of starting material and input amounts. So with that being said, I'd like to move on to the first component of our three-piece solution for gene profiling. And the first step is to work through the labeling or the assay itself, the biochemistry. So for the Human Transcriptome Array, which is what we'll be talking about, focusing our time on today, there are really two options for target preparation from Affymetrix. We have the Affymetrix WT Plus Expression Kit, which is really just our standard whole transcript or WT assay that's to be used with intact, high-quality RNA. And there's a broad range of starting input amounts that you can use with this kit, starting as low as 50 nanograms and going all the way up to 500 nanograms. The second option for target preparation is our Affymetrix Sensation Plus FFPE amplification and WT labeling kit. Now this kit is specifically designed to be used with degraded samples, which are derived from formalin-fixed, paraffin-embedded sources. And you can start with as little as 20 nanograms, so very little starting material to generate really high-quality results. And I'll show you some information a little bit later about the correlation between fresh frozen and FFPE samples. And of course, as with all of our reagent kits and arrays, we have very high technical reproducibility when you're going from lot to lot. So you don't have to worry about making sure that you have one lot to do your entire study. You can separate out your study, and as long as you're using the same reagent kit, you can expect very high technical reproducibility from our reagent kits and our arrays. So let's dive just a little bit deeper into each of these reagent kits. We'll first start with the WT Plus reagent kit. And this kit is designed to be easy to use and to deliver excellent reproducible results. With this kit, we don't require any rRNA or ribosomal RNA reduction or poly-A selection steps. So what this means is it saves time and money. It ensures that the entire transcriptome is profiled, so you're not excluding any transcripts by doing any of these reduction steps. And personally, when I'm working with RNA on the bench, I want to do as little manipulations to that RNA as possible. So if I can avoid having to do RNA, ribosomal RNA or poly-A selection, that's one less step that I have to subject the RNA to, and it just means a little bit higher quality results at the end of the day. And again, we touched on the low input amounts for this particular kit. Use as little as 50 nanograms of total RNA. 100 nanograms is recommended if you have it, and you can go all the way up to 500 nanograms. And I'm going to show you a little bit of information that talks about different starting amounts and how reproducible the results are. Now, the great thing about the WT Plus kit is that it can be used with multiple different sample sources. So you can use this kit with RNA derived from tissue, from cells, from blood, and from other sample sources. And if you have specific ones, please let us know, and we can work with you to make sure that that will work properly. So this slide here is a bit of an eye chart. I don't want to go through the whole thing. What I do want to mention is that each row is MAQCA and MAQCB are different, two different control RNAs that were used in the microRNA Quality Control Consortium study that was done several years ago, funded by the FDA. So these are reference RNAs that are being used, and you can see that we have input amounts at 50 nanograms, 100 nanograms, and 500 nanograms. And what I want to direct your attention to is some of the quality control metrics that we normally or generally use to check the quality of the array data across these different concentration or input amounts are very similar. So if you look at things like percent present, basically signal to noise, and also this thing called positive versus negative area under the curve, which is a measure of sensitivity and specificity, you can see that the arrays, that the quality control results are very consistent across a broad range of input amounts. So whether you start with 50 nanograms or 500 nanograms, you can be sure that you're going to get very high quality results. And I alluded to this just a little bit before. So these are signal and fold change correlations, both at the gene level and the exon level, across comparing different input amounts. So up here in the top left, we're looking at gene level at 50 nanograms versus 100 nanograms. And you can see that for within both MAQCA and MAQCB, the Pearson correlations are quite high at 0.98. And also between the two, the fold change correlations are also quite high. This works as well at the gene level at 100 nanograms versus 500 nanograms, and it extrapolates also very well to the exon level. And we'll talk about why looking at exon level data is important a little bit later in the talk. But I wanted to point out that regardless of the input amount, 50 nanograms, 100 nanograms, or 500 nanograms, the correlations are very high at both the gene and the exon level. So you can be assured that no matter what input amount you're using, from 50 nanograms all the way to 500 nanograms, that you're going to end up with very high quality data at the end. Okay, so WT plus should be used with high quality intact RNA. So let's switch gears a little bit, and we'll go to the second reagent option, or the second assay option, which we call Sensation Plus. Sensation Plus is designed to work with FFPE, or formalin-fixed paraffin-embedded derived RNA. So again, as with the WT plus, Sensation Plus requires no ribosomal RNA reduction or poly-A selection. So again, it saves time and money and allows you to do fewer manipulations to your RNA to make sure that you get the highest quality results at the end. Input amounts, very low for the Sensation Plus kit. You can go as low as 20 nanograms. If you have 50 nanograms, the percent of success, or your success rate per sample, goes up just a little bit. So the quality of the data will be the same whether it's 20 nanograms, 50 nanograms, or 100 nanograms input amount. It's just the success rate, the conversion rate for each of your samples is a little bit higher if you can start with 50 nanograms versus 20 nanograms. And Sensation Plus, as with WT Plus, was designed by Affymetrix, by our product development scientists in our Cleveland facility. So it's a proprietary chemistry, and it's designed specifically to work with the Affymetrix arrays to reduce background and produce the best possible data. So let's look at a little bit of the data here. So what we're looking at are Pearson correlations. This is at the gene level, FFPE versus fresh frozen tissue. And we have 3 cohorts here. So there's a 9-year-old set, normal and tumor. There's an 8-year-old set, normal versus tumor, and a 7-year-old set, normal versus tumor. And you can see that the correlations are very high. So this is fresh frozen versus FFPE-derived RNA. And the correlations between fresh frozen and FFPE are very high. They're always greater than 0.9. And as you can see, they actually get a little bit better the older the samples get. Our kit tends to work very well with older samples, as old as 10 years old. Our kit will work very well with. So I want to take that just one step further. We took the same data set that we looked at in the previous slide. This is a colon cancer data set, and ran the results through the Ingenuity iReport pathway. And so what we're looking at here on the x-axis are disease types, or sort of bins of disease. So you have digestive organ tumor and colorectal tumor, intestinal cancer. So these make sense with our colon cancer data set. And on the y-axis are the numbers of differentially expressed genes which fit into each of these disease categories. And what I want to direct your attention to is that the blue bars are fresh frozen, and the green bars are FFPE derived RNA. And what you can see is that for each of the disease categories, the fresh frozen and the FFPE samples are performing almost identically. So you'll see that there's the same number of genes in each of the categories. There's a little bit of differences, which you would expect, especially using older samples. So these are 7-year-old samples. So you would expect to see those differences, but in general, the results are almost identical between fresh frozen and FFPE. So what you can expect is that when you use the Sensation Plus kit with your FFPE-derived samples, that you're going to get very high-quality data at the end. Okay, so that's the first step in our complete solution. So we've talked about the assay, the biochemistry. So now let's switch and go into the second portion of our complete solution, which is the array itself. And we call this the Human Transcriptome Array, or the HTA 2.0. So I want to first start with talking a little bit about why it's important to look not only at the gene level, so gene level differential expression, but also looking at alternative splicing. So we know that there are tens of thousands of human genes, but those tens of thousands of human genes code for hundreds of thousands of proteins. So it's not possible to have a one-to-one relationship between gene and protein. And the main factor that contributes to this is alternative splicing. And we're finding more and more, we're seeing more and more publications coming out, that alternative splicing is one of the most critical factors when you're looking at the complexity of the human transcriptome. So it's really important not only to look at the gene-level differences, but also the exon-level differences so that we can see these alternative splicing events. And here is a paper that we just saw, just got published actually this month, October 2013, in Cancer Discovery. It's a research brief. And what we're seeing here is this particular investigator looked at uveal melanoma, which is an eye malignancy. And they looked at RNA-Seq, Sanger sequencing, and also looked at HTA information, so data derived from our Human Transcriptome Array. And they found that alternative splicing was one of the major contributors to the prognosis that's associated with this particular disease. And they saw some changes, some alternative splicing events in the human transcriptome data that actually was missed in the RNA-Seq data. And we'll talk a little bit more in depth about that further down the line. But I just wanted to show you something hot off the press, just came out this month. So we're seeing that this is really important, and it's important to look at both levels of differential expression when you're doing your research. So here is just a basic example of why alternative splicing is important. So we're looking at the BCLX gene, and BCLX codes for 2 isoforms. There's a long isoform, which is on the left-hand side of the screen, and a short isoform. And the only difference is the cassette exon in the middle. The long isoform, that exon is retained, and in the short isoform, that exon is spliced out. And in the long isoform, we're actually seeing that those cells that express that particular isoform, the cells are thriving perfectly well. And the cells that are expressing the shorter isoform are actually dying. So if you were looking at just gene-level information, you'd potentially miss something that was telling you why these 2 different cell populations were behaving very differently. Some cells were living and some cells were dying, and it's a result of alternative splicing rather than whole gene differential expression. So you really need to know which isoforms are being expressed in order to get a complete biological understanding of your system. Okay, so now let's start talking more specifically about the Human Transcriptome Array. So the Human Transcriptome Array, the current version that's commercialized by Affymetrix is an offshoot of a version that was designed by Ron Davis in Stanford, California. And so this is the paper was published in PNAS in 2011, and we'd be happy to send you a link to download this paper. I encourage you to read it. This is the HTA version 1, the first version of the Human Transcriptome Array. We made some modifications to it, and now HTA 8 version 2 is the one that's being commercialized and available to you. So let's look just a little bit about the evolution of the expression arrays from Affymetrix. So this slide shows a hypothetical genomic locus up here at the top, and this locus comprises obviously exons and intronic regions. And we can generate 3 isoforms from this particular genomic locus. So if we looked at the 3' IVT array, this is our older generation arrays. The probes here, which are denoted by these little hash lines here under each of the colored bars, indicate the probes that interrogate, that we have on our array that interrogate this particular genomic locus. And you can see that on the 3' arrays, these probes are all situated at the 3' end of the gene. So if we had any sort of alternative splicing that was happening 5' to that, we potentially missed those events. In 2008 or so, we came out with our first generation of whole transcript arrays, which we termed the gene or the exon arrays, which I'm sure many of you are familiar with. So instead now of just putting the probes down to the 3' end of the gene, now we actually tile probes against each individual exon. So this would be across the entire transcript. So this is great. Now we can have more robust information at the gene level, and we can start to look at exon level information as well. But with the Human Transcriptome Array, we'll take this one step further. So not only are we going to increase the density of the probes that interrogate each exon, so we have on the exon array, which is the first generation whole transcript arrays, we have about 4 probes that interrogate each exon. Now with the Human Transcriptome Array, we have 10 probes that interrogate each exon. So not only are we getting more robust exon level signal, but we've also included these spliced junction probes as well. So wherever we have a known spliced junction, we tile 4 probes down to that spliced junction. So now we can combine exon level information with the splice level information and start to get a much more complete view of the alternative splicing that's happening. So I'm going to walk you through this really quickly. So if we take this first isoform here up at the top, we can see that we have this exon number 1, exon number 2 is spliced out, and then exon number 3 is retained. So what we'd expect to see on the Human Transcriptome is signal from the probe set that interrogates exon number 1. So we'd have signal coming from this probe set. We'd have signal coming from the probe set that interrogates exon number 3, but no signal from the probe set that interrogates exon number 2 because that exon is being spliced out. That exon is not being retained, so we wouldn't expect to get any signal. So there's our first level of information. That's telling us, okay, there's something going on here at the exon level that I might want to look at. Now there's 2 more levels of information that we can look at to make sure that this splicing event is actually truly happening. So because exon 2 is no longer present, there won't be a junction between exon 1 and exon 2. And the same thing for the junction between exon 2 and exon 3. With no exon 2, there's no junction between exon 2 and 3. So these right down here are the probe sets that are interrogating the junction between exon 1 and exon 2. So we'd expect no signal from those probe sets, and also we'd expect no signal from the probe set that interrogates the junction between exon 2 and exon 3. So there's 2 more pieces of evidence. But we go one step further, and because this is a known isoform, we know that there is a junction between exon 1 and exon 3. So we tile down 4 more probe sets that interrogate the junction between exons 1 and exon 3. So this probe set we will actually get signal from, and that's our 4th piece of evidence telling us that there is an alternative splicing event happening at this particular location. We'll go more in detail on that. I'm going to show you a little bit of data, but I wanted to give you an introduction first. So before we get into the actual alternative splicing analysis, I wanted to tell you what's on the array. I don't want to go through all these numbers, but suffice to say that we break the content on the HTA array down between protein coding and non-protein coding content. So the protein coding content are the things that are interrogating the protein coding regions, the genic regions. So everybody asks me, well, we say we interrogate 44,700 genes. These are really what we call transcript clusters. We know that there are about 25,000 or 26,000 genes in the human transcriptome, but some of these things are made up of, we have ESTs. There could be different isoforms that are being expressed, and we sort of encapsulate all those into this gene category. So that's why we come up with this number, 44,700 genes or transcript clusters that we're interrogating. So then we have 245,000 transcripts. These are the isoforms that are being expressed. So these are all of the known non-redundant isoforms that we know exist. And so those are all of the isoforms that are being interrogated on the array. And those are all comprised of about 470,000 exon-level probe sets. So these are the probe sets that are interrogating right at the exon level. So that this is what allows us to look at the alternative splicing events. And we have another 340,000 splice junction probes as well. And then we also have some non-protein coding content. So these would be long non-coding intergenic RNAs or link RNAs. We have about 23,000 of those, and then about another 100, and those are comprised of about 103,000 exon-level probe sets. What's really interesting is that the content is derived from several public databases. So all of our content is taken from the public domain, so none of this is proprietary information. And we do that because we want you to be able to find your interesting results using the array and then link out to these public data sources and get more information about the things that you see which might be interesting in your study. So the content's derived from multiple sources, including RefSeq, Ensemble, UCSC, Vega, the MGC, and then we have the long non-coding content is coming from non-code, LinkRNA-DB, and the Human Body Map, LinkRNA, and TUP catalog. So what does that all mean? Well, we have multiple. This is a single gene. I don't remember which gene it is, but this is a single gene, and we can see that there are multiple isoforms that are known for this particular gene. So that's what all of these blue transcripts represent. These are all the known isoforms. Now, on the left-hand column is the data source that known isoform comes from. So this first one comes from Ensemble. The second one is in both RefSeq and UCG. So you can see that all of these known isoforms are from multiple different data sources. So if you were looking at a single data source or a single database, you might see only one or two of these isoforms, but potentially miss the isoforms that are in the different databases. So what Affymetrix has done is we've compressed all of these databases into a single comprehensive database that's available right within our software, right within Transcriptome Analysis Console. So you have access to all of these multiple databases all at once just by running the analysis in our software. It's very powerful to be able to have all of this information in one single location rather than having to cross-reference multiple databases several times. It really speeds up your analysis. And also, I don't want to leave out LinkRNA. We tend to skip over this, but it's really important. And in fact, the paper that I discussed about the eye malignancies just a few minutes ago, they found that alternative splicing was also happening in a long intergenic non-coding RNA, which is an important regulatory factor in cancergenesis. So it's important not only to look at the coding regions, but also the non-coding RNAs. These become very important regulatory mechanisms, and they also have a very big effect in transcriptional modification. So we're seeing that these become very important in things like cancer research. And just to kind of drive the point home, there's a ton of publications that are coming out that are referencing non-coding RNAs. So don't just look at coding RNAs, but look at both non-coding and your coding RNAs at the same time using the same array. So we've already talked about this. I don't want to spend too much time, but I just want to reiterate that there are 4 pieces of evidence when we're looking at an alternative splicing event. There's the exon of interest, so whether this exon is spliced in or spliced out will tell us whether we have signal from the probe set that interrogates that particular exon or not. Then we can look at the 2 junction probe sets that actually interrogate the junction between the neighboring exons, and finally we can look at the exclusion junction probe set. So this is the splice junction probe set that interrogates the junction between, in this instance, exons A and C, and that's the fourth piece of evidence that we're looking at when we look at alternative splicing events. So here's a figure from the Ron Davis paper. So this was done on HTA version 1. We have since validated, and you get the same results on HTA version 2, which is the commercial version of the array. But I wanted to point out something interesting that they were able to do with their study. So their study looked at liver versus muscle, okay? And they know that there is one gene, or there actually are several genes, but this figure illustrates one gene called SLK that they know exhibits differential isoform expression between the two sample types, so between muscle and liver. So if we look at, on the heat map, if we look down the diagonal, this is the actual exon-level expression. So this is the expression for exon 12. This next box on the diagonal is the expression for exon 13, 14, 15, 16, and so on and so forth. As you get towards the red end of the spectrum, that's high expression for that particular exon. And if you go towards white, if you get towards the white color, then that's a low level of expression for that particular exon. So you can see in the liver samples, most of the exons are expressed at a relatively high level. But if you look in muscle, as we go down, if we look at exon 15 in this heat map is white, meaning that there's very little expression or no expression at all for that particular exon. So that's piece of evidence number one, indicating that we might have some sort of alternative splicing event happening there. So exon 15 might not be expressed in the muscle samples. Now, if we look off the diagonal, so we go from here to here, this is the exclusion junction probe set that goes from exon 14 to exon 16. So now we're skipping exon 15. So this is the splice junction probe set that skips exon 15. And you can see that there is some expression from that splice junction probe set. So now we have two pieces of evidence to back this alternative splicing event up. There's no exon-level signal for exon 15, and we also have some signal coming from the probe set that interrogates the junction between exons 14 and exon 16. Now, if we go back to the liver samples for a second, I said that we do have expression from exon 15 in the liver samples. So that's what they expected. They expected to see this differential exon-level expression between the two sample types. Now, what they didn't know is that actually both isoforms are expressed in the liver sample. So this is something novel that they found by using the Human Transcriptome Array. So if you look closely, exon 15 is not expressed at as high a level as some of the other exons. It's not quite as red. And also, if you look at the splice junction probe set that's interrogating exons, the junction between exon 14 and exon 16, you can see that there is some expression from that splice junction probe set as well. So what this tells us is that both isoforms are being expressed, the isoform that includes exon 15 as well as the isoform that skips exon 15. And this is something completely new that they were able to derive by looking at the data from the Human Transcriptome Array. Okay, so I'm going to switch gears a little bit, and I want to talk a little bit about why we want to look at exon level and what's maybe the best way to do that. So this is a relatively old figure. This came from the publication Cell in 1976. So the numbers are a little bit off, but the general trends actually hold true. So if you took a tube of total RNA, about 40% of the total RNA in that tube would be comprised of only 10 genes. And those are the 10 genes that are very highly expressed. We're talking about things like maybe our ribosomal RNA, some of the cell scaffolding genes that code for cell scaffolding proteins, things like that that are very, very highly expressed in most cells. So that's almost half of your total RNA made up of only 10 different genes. Another 45% of that tube of total RNA is made up of just 600 more genes. So these, again, are very, very highly expressed genes in your cell. So 85% of that tube of total RNA is made up of just over 600 genes or so. And it's going to be a little bit different depending on sample types. So it may be made up of 1,000 genes or 500 genes, but suffice to say that about 85% of that tube of total RNA is comprised of a very small number of genes, which leaves the rest, the remaining 15% of your total RNA, which is all of the other genes in the human transcriptome. Excuse me. So what that means is that the rest of these genes are in a relatively low abundance in your sample versus the other genes that are expressed at a very high level. Those would be at a very high abundance. And what we're finding is that a lot of the interesting biology is being found now in these low abundance transcripts, these transcripts that are expressed at a very low level and can have a very profound effect on your biological system. So what I want to show you is just a little bit of a comparison between our human transcriptome array and some RNA-Seq or next-generation sequencing data. So I'll orient you to the figures, and then we're going to have two slides that talk both one at the gene level, which is this slide, and then the next slide will talk more specifically about exon-level expression. So the x-axis represents relative abundance, and here on this particular plot, we're using RPKM, which is a typical measurement of abundance used by the RNA sequencing community. So high up on the x-axis means very high abundance transcripts. Low on the x-axis means very low abundance transcripts. Now the y-axis represents coefficient of variation, so %CV. So the higher the CV, the more noisy or the more variable your results are, meaning that you get lower quality results. Now the red or the pink line here indicates the CVs for the human transcriptome array. And because we're a hybridization-based technology, abundance doesn't really matter. As long as we're above our detection threshold for our hybridization and our detection chemistry, you'll be able to detect that particular transcript. So whether it's at a high abundance or a low abundance, we can detect that transcript. And the CVs are basically flat across this broad abundance range. Now, if you look at RNA-Seq data, for instance, if you're running, I think this is 1/16 of a high-seq lane, so you're looking at about 15 million mapped reads. At the high abundance transcripts, you're getting very low CVs, so very good data. But as you start to look at the lower and lower abundance transcripts, which is where that interesting biology is happening, your CVs start to go up, meaning that your data quality starts to degrade a little bit. Now, you can mitigate that. You can counteract that by running deeper sequencing. So generating more mapped reads per sample has the effect of lowering the CVs, especially in these low abundance transcripts. So you can see as we go from 15 million to 30 million to 60 million mapped reads, the CVs start to decrease, which is great. So if you're sequencing deep enough for your particular study, then you should get great data. And I want to preface this by saying that I think RNA-Seq is a fantastic technology. It's just we have to make sure that you're sequencing deep enough to get the proper quality of data that you're looking for. So again, I just want to reiterate that we're finding that a lot of the interesting biology is happening in these low abundance transcripts, these transcripts that are expressed at very low levels. So here's the same type of plot, but now we're looking at exon level data, and you can see that the y-axis has changed quite dramatically. So the percent CVs now go up to about 200%, whereas on the gene level plots, they went up to, I don't know, 75 or 100 or so. So the same trends tend to hold true because, again, the arrays are a hybridization-based technology. You can see that the CVs are relatively flat from very high abundance transcripts all the way down to low abundance transcripts. But again, if you're sequencing at about 15 million mapped reads, which means you're multiplexing 16 samples on a lane, typically on a high-seq, then your CVs for these low abundance exons are quite high. So as you sequence deeper and deeper, then your CVs start to come way down. Okay. Just one more note. We talk, sometimes we hear people talking about coverage. We do it too. We cover a gene or an exon with a particular number of probe sets. Here we're looking at coverage from an RNA-Seq point of view. So for gene coverage, if we're looking at 10x gene coverage, that means I need to have at least 10 reads that hit this particular gene. So they could be anywhere throughout the gene. They don't have to hit any individual exons. So we just need to make sure that we have 10 reads that hit this particular gene. So that's 10x gene coverage. But the difference here is if we talk about 10x exon coverage, and we're focusing a lot on exon because, again, the alternative splicing, looking at those differently expressed isoforms, is where a lot of the interesting biology is happening. So if you want to get exon-level information, if you need 10x coverage, that means you need to hit each exon 10 times. So look at the depth of coverage or the amount of reads that you need to get 10x exon coverage versus 10x gene coverage. It's quite a bit more. Okay, so we've talked about the assay. We've talked about the array itself. So let's talk about the third piece of the puzzle, which is the actual data analysis itself. I love this slide because no matter what technology you're using, what platform you're using, you run your assay, you generate your data, and then you sort of end up with this pile of data on your desk, and you're looking at it and you're like, okay, where do I find the corner piece? Where do I start? And so with the Human Transcriptome Array, Affymetrix has launched our own software called Transcriptome Analysis Console, which allows you to do a statistical analysis at both the gene level and the alternative splicing level for your Human Transcriptome Arrays. This software works in conjunction with the Expression Console, which a lot of you may be already familiar with. So you'll need both Expression Console and then Transcriptome Analysis Console in order to process this workflow. You can do the analysis at different levels, whether you want to look at gene level or exon or alternative splicing level. And TAC is really, or Transcriptome Analysis Console, is really the first time that Affymetrix has designed expression software to do statistical analysis. And we're really happy with the results, and we're getting some really good feedback at the ease of use and the power behind the software. So software is really, really easy and really intuitive to use. First, you start by specifying the type of analysis that you want to perform. Do I want to do a gene level analysis? Do I want to do my analysis at the alt splicing level, the exon level? So you select your level of analysis, and then you'll import your chip files, which are the output from Expression Console, into Transcriptome Analysis Console. Once your chip files or your raw data are imported into Transcriptome Analysis Console, you'll separate them out by condition. So if we're doing liver versus muscle, I'll take all the liver chip files and put them into one condition, and take all the muscle files and put them into the second condition. Then you can customize this a little bit by changing the name of the condition so that you remember how the analysis was done, and you hit the Run Analysis button, and it will perform a standard sort of ANOVA statistical analysis for you. So let's look a little bit at the gene level. So I just wanted to show you, you import your chip files here into the top window and then drag them into your particular condition of interest, and then hit the Run Analysis button, and it will run the analysis for you. The software is not limited to only 2 conditions. You can do 3 conditions, 4 conditions. You can add multiple conditions, and it will do, all of the pairwise analyses for you. And so here's what the results look like. You'll end up with a table view, which will contain the probe set ID or a transcript cluster ID, and then lots of information about the analysis itself, including things like p-value, fold change. You can do a FDR-adjusted p-value, and then it will give you also some annotation information, so things like gene symbol and a description of what that particular entity represents. Now you can sort on this table. You can sort on fold change. Say I only want to look at things that are greater than a fold change of 2 or greater than a fold change of 3. You can sort right in the table, and all of the resulting plots that we can generate will update dynamically based on that sorting. So down here in the bottom left is a scatter plot. So essentially we're looking at signal of one condition on the x-axis versus the signal of the second condition on the y-axis. So what we'd want to look for in a typical study where we're looking for differential expression between two conditions is we want to see things that are very highly expressed in one condition but not very highly expressed in the other condition. So things that might be in the upper left corner or in the lower right corner of these scatter plots. And you can see the things in the middle are grayed out because those don't pass the filtering that I set in the table. So I said I want to exclude anything that isn't greater than a 2-fold change. And so all the gray area, all the gray probe sets in the middle are being excluded because they don't pass that particular threshold. We can also look at a Volcano plot. So what we're looking at in the Volcano plot is fold change on the x-axis and something we call significance, which is just a log transformed p-value on the y-axis. So this gives you just an added layer of information. So not only can we look at just fold change, so is it really upregulated in one condition versus another, but also do we have a very significant p-value as a result of our statistical test as well. So if something was very highly differentially regulated but didn't have a very high p-value, then that might not be something you'd want to follow up on. And we can look at that very easily in the Volcano plot. And finally, we can do, at the gene level, we can do some hierarchical clustering as well. So if you wanted to generate a signature that helped you separate two subtypes of cancer or liver versus muscle or something like that, you can generate a gene list and then cluster those genes into the two categories, and that will help you discern a signature that can differentiate those two conditions from each other. Now, I also wanted to point out that you can select by lasso some of the important hits in these plots that you're looking at. So I just draw a lasso around them. They all get selected. They get highlighted in the table view, and then I can link out to external databases and get more information. So you can actually right-click on any of these entities and go out and search Ensemble or RefSeq or get genomic context by looking at the UCSC genome browser. So the software is not only intuitive, but it allows you to link out to external references as well to try to get much more information about what those particular hits are. So that's the gene level analysis. Let's take a look quickly at the exon or alt splicing level analysis as well. So this is done, again, through the same piece of software. You use Transcriptome Analysis Console. The results look a little bit different. We get a table view. Now we have each of the transcript clusters, which is sort of like a gene. They're all grouped together. And then within that, now we'll have each individual exon level or splice junction probe set listed that belongs to each particular gene. And again, we'll have things like fold change, p-values, and this other thing called splicing index, which is sort of like a exon level fold change, but we do a gene level normalization first. So it takes care of any gene level differences before we look at the exon level differences. And you can sort and filter based on any of these columns, just like we did with the gene level analysis. And then we also have a really great visualization tool that's built into Transcriptome Analysis Console that allows you to see your interesting hits and then find out whether there are known isoforms that express or that look like the isoform that you're seeing in your particular sample. So let's look at one example of how you would see an alternative splicing event by using the Transcriptome Analysis Console software. So this particular example comes from the liver versus muscle data set, and that data set is publicly available on our website. You can download it and do the analysis yourself and confirm these results right on your own laptop in your office. So we're looking at the ANXA7 gene, and we know that there are two isoforms that are expressed. One isoform is expressed in muscle, and the second isoform is expressed in liver, among other tissues. And the question is, can we determine if there are any expression changes, whether they be at the gene level or at the alt splice level, by using the TAC software? So first, we'll look at the gene level. And we find, once we do the analysis, that the ANXA7 falls sort of right here in our Volcano plot. So it's not very highly differentially regulated at the gene level. The fold change is very, very small. And even if it was, the p-value, so if we look at the significance value, it's fairly low on the significance axis. So at the gene level, this isn't a very interesting hit. So if you were doing your analysis solely at the gene level, you actually may miss this particular gene, this ANXA7 gene, because it's not interesting at the gene level. But in fact, at the exon level, this is where the interesting biology is happening, and this is where we need to drill down to get to this information. So what I want to do is draw your attention to this third plot down, or this third line down here at the top. This is the information that we call splicing index. So essentially, again, this is the fold change information at the exon level that's normalized first for differences at the gene level. So we take the differences at the gene level out first, and then we look at the exon level differences. Now, if we look at our legend down here at the bottom, the green means that the splicing index, or the fold change, is downregulated in liver versus muscle, and red would indicate that it's upregulated in liver versus muscle. So we can see that for this particular exon, this exon is downregulated in liver versus muscle. Okay, so that's the exon level information. Now let's look at our other forms of evidence for this alternative splicing event. So the inclusion junction probe set, so that means the junction probe set that goes from this neighboring exon to the exon in question, is also downregulated. It's downregulated on this side, and it's also downregulated on this side, meaning that these splice junctions don't exist in this particular sample. And our fourth piece of evidence, this exclusion junction probe set, that means we go from this exon over here, we skip the exon in question and go to the third exon. That junction probe set is actually being upregulated. So here's our four pieces of evidence that we were talking about before in a real-life example. The exon level expression is downregulated. The inclusion junction probe sets are also downregulated, but the exclusion junction probe set is upregulated. So now we have four pieces of evidence telling us that there is some sort of alternative splicing event going on at this particular location. Now, if you don't believe me, you can look at the actual signal from each condition separately. So this is the liver signal here at the top, and this is the muscle signal here at the bottom. The yellow indicates that it's downregulated, and the blue indicates that it's upregulated or being highly expressed. And you can see that in liver, this exon is not being expressed at a very high level, and the junction probe sets are telling us the same story. And you can see in muscle that we are expressing this exon at a high level. So the aggregate information is telling us the same thing. It's actually pulling all this information together and giving us a very easy, user-friendly way to spot this alternative splicing event. Okay, so a quick summary over the software itself. So again, the software is called Transcriptome Analysis Console, designed to be really easy to use, but it's very powerful under the hood. We're using very common statistical models to generate the results. The analysis is very quick, and you can do it at the gene level, the exon level, and the alt splicing level, all within the same software. You can filter your data, and there's lots of visualization options that allow you to see what you're looking for, and you can change and sort of alter the way you're looking at your data so that you can see just what you want to find. And then we've also provided the functionality to link out to the external databases so that you can get more reference information, and also you can confirm that these hits that you found, say you found an alternative splicing event, link out to a paper in the RefSeq database, or in PubMed or something like that, that references that, and you can confirm that that splicing event has been seen by another researcher. Very quick, very easy. Now, there are some minimum requirements to run Transcriptome Analysis Console, the biggest of which is that you need a 64-bit computer. Now, most computers these days are 64-bit. If you don't have one, you can try to collaborate with somebody that may have one. Check with your core facilities and things like that. A lot of times they'll have a computer as well that you can have access to. Now, the greatest part of all of TAC is that it's free. There's no charge for the software. You can go onto our website right now and download the software and start playing around with it. So I just wanted to kind of end with this slide here showing that the time to results. Remember, one of the factors that we talked about very early on in the presentation today was time to results, getting to your results very quickly. And the time to results with the array analysis is very, very quick. It's less than 1 week. And that's processing again, this process is very scalable, so you can process multiple arrays at the same time, and they'll all take, you get all of your data at once, and you can analyze that data and have your preliminary results in about a week's time. And I think, I won't belabor the point, but I think we compare very favorably with some of the competing technologies that are out there right now. So in conclusion, we're offering a complete solution to transcript profiling. We have the assay itself, so there's two options, WT plus or Sensation plus. It's very quick, efficient target preparation, less than 4 days from RNA to results, works with lots of different sample types, fresh, frozen, FFPE, blood, and low sample input. So you can keep your precious samples, run your results on the HTA, generate your results on the HTA, and then validate with that same sample type. So you're not using all of your sample just in the initial data generation. So that's piece one. Piece one is the assay. The second piece to the complete solution is the array, the Human Transcriptome 2.0 array. We spent a lot of time talking about that today. Whole genome or whole transcriptome coverage, all known genes in the public domain are covered at a very high level of density, so 10 probes per exon, plus those splice junction probe sets. And we include non-coding content. So not only are you getting the coding information, but you're also getting the non-coding content at the same time. And then the final piece, the third piece to this complete solution, is the analysis component. We have software that's freely available, Expression Console and Transcriptome Analysis Console. They work in conjunction with each other and allow for the detection of not only differentially expressed genes, but now we can really dig in and look at exon-level changes and start to look at alternative splicing events and which isoforms are being expressed in your particular studies. The analysis is really quick, really easy, and can be done right in your office. You don't need a supercomputer to do the analysis. As long as you have a 64-bit computer, you're good to go. You can do it right in your office, so you don't have to spend time and money collaborating with folks that have the more intense computing resources.